then we might as well stop it. Uh, I can. Thank you. एक मिनट नहीं नहीं जाने के लिए Okay, shall we start? So my slides are visible online. I'm guessing they are. Yeah. So, okay. Good morning, all of you. My name is Tej, and Sondata introduced. I work in the field of genomics, and today's talk is going to be a brief overview of what all aspects of genomics are being helped or revolutionized by AI. Okay. So before I start, I would like to give a very brief introduction about what I do or how I've ended up doing what I do. So genomics can be pretty much considered a subsection of what is known as bioinformatics, which is a interdisciplinary field which brings together biology or healthcare and big data and computer analysis. So which means that you need to have understanding of two different fields. But that doesn't mean that I started out as a computer science graduate. In fact, my master's was in zoology and my PhD was in biochemistry. And bar for the bit of interest I had on the side for computer programming, I have not done anything computer related during my PhD. It's after my PhD that I decided that I want to, I've noticed how research was becoming increasingly high throughput, which means that you can't analyze all that high throughput data just by looking at it one by one. You will need help of computers. That's when I shifted my focus to informatics. And now I basically use a lot of computer assistance to look at my data, understand what it is saying, and that's the kind of work that we do. Okay. So in this talk, so this is how the talk is going to be structured, where I'll talk about 10 minutes about genomics and what it is, how it sort of works. And at any point of the talk, it's okay if you do not fully understand the technical know-how of how things are working. What I want you to do is have these key take-home messages with you so that you know that on an overall scale, what is happening with a given section. Okay. So after we talk about what genomics is, I'll cover three subsections on where AI is helping this field. One is in sequencing and variant analysis, and then in cancer genomics, and also in clinical genomics. And we will very briefly towards the end discuss what kind of challenges are there in this and what can be done. A brief discussion over that. Okay, so going to the introduction, we know that DNA is the genetic material for pretty much all the species, all the living organisms, except for a few RNA viruses, which use RNA as genetic material. Most of the other, or pretty much everything else, use DNA as its genetic material, which is also akin to be considered the instruction manual for life, right? So all the processes that we see in a cell or an organism pretty much has some connection to the underlying blueprint, which is DNA, right? So to understand how DNA functions, what we need to do is study how these things, how DNA sequences differ from each other. We can do this at a species level, where we know that there can be a lot of similarities between species, but uh, we can also study this at individual level. If you, let's say, take different people, or let's say of humans, then we are up to 99.9% in our DNA content, right? And we can also study this at cell level, where we might have the same DNA sequence, but these cells do different functions. Okay, but more than similarity, what we focus on is differences. Because if, of course, 99.9% of DNA is similar between people, but it's that 0.1% which makes us the unique us, right? We are all different in our own unique way. And I'm not talking just, of course, it's also interesting to study just the basic phenotypes, such as, let's say, height, or whether you have curly hair and stuff like that, or eye color. But it's also important to understand how these differences lead to disparities in health, right? Someone might be more prone to getting an illness, let's say cardiovascular disease or cancer or whatever it is. And focusing on these differences in DNA sequences helps us link these differences to various disorders. And that's where we are mostly interested in. So what is genomics then? So a genome is nothing but a collection of the entire genetic material present in a cell or a tissue or an organism, right? So to give an example, the human genome is 3 billion bases long, or 3 gigabases. So this GBP is gigabases, just like how we have gigabytes in computer. So 3 gigabases is 3 into 10 power 9 base pairs, right? And the study of genomes is called as genomics. Okay. So like I said, genomics begins by understanding what the DNA sequence is. So the underlying technology to study any of this is DNA sequencing itself. And in the next couple of minutes, I'll briefly cover how this was first introduced and how it was upgraded. So the most commonly used method is called Sanger sequencing. Many of you might have actually looked at that kind of a data. And the way it works, assuming you all know how DNA replication works. So what you do is you take the DNA sequence that is of interest to you, and you put it in, let's say, 4 tubes, where you add each of the, the dinucleotide, or DNTPs, dinucleotide triphosphate, and you specifically add something called as dideoxynucleotides, which terminate every time that DDNTP gets added. And when you take each of those 4 reactions and resolve it on a gel, what happens is that you will see fragments that correspond to a specific nucleotide line up like this, and you can read the sequence this way. Again, don't worry if you're not getting the clear concept of how this is done. But the point is that this was first developed by a gentleman named Frederick Sanger, for which he has won Nobel Prize in Chemistry in 1980. Right? And currently, we don't use those 4 different tubes. What we do is we use fluorophore embedded DNTPs to resolve them in one go using laser emissions or fluorescence, and we get like this. Again, if some of you have worked with Sanger sequencing data, you would have seen this kind of chromatograms, which can be converted back to like, let's say, yellow color fluorescence at the bottom of the peak means it's a G, and based on color you can deduce what nucleotide was present. And using this kind of a method, Frederick Sanger had sequenced the human back in 80s, which looks something like. But the human genome, like we discussed, is 3 billion bases long. So you can't sit, which means it's roughly 2 million times larger than this. So you can't sit and do 2 million of such reactions, right? Or that's what you would think. But the Human Genome Project did exactly that. So where they have actually had hundreds and hundreds of these sequencers running for over 15 years with a cost of $3 billion. And they've generated millions of such fragments, and they've put together the entire human genome, which was first released in the year 2001. But at that time, there were roughly 8% of the human genome was not covered. These were all chromosome ends or other difficult to map regions, which was not covered. And 20 years later, in 2021, was when the first complete human genome was sequenced. Okay. So, but as you can see, this took 15 years to sequence one genome. But these days, we sequence several, like, for example, the sequencer that we have can sequence 30 genomes in one day. Okay, so that's how much the technology has progressed. And with that technology progress, there was also a drop in the associated sequencing cost. So as you can see, this is the amount of money needed to sequence one human genome. It started with $3 billion for the Human Genome Project, but right now, in under $1,000 or 80,000 rupees, you can sequence a full human genome. Okay, and as you can see, there is a sharp decline here, right? And that's the advent of something called as next generation sequencing. Okay, so next generation sequencing is nothing but a much more high throughput version of Sanger sequencing. So where you are not sequencing one fragment at a time or even 96 fragments at a time, which is what is the current capacity, but you're sequencing billions of DNA fragments in a single go. Okay. And this is one of the NGS machines called the Illumina NovaSeq X Plus, and this can sequence something like 80 human genomes in one day. So that's how much the technology has progressed. Okay. So, and because it has become faster, cheaper, and much more high throughput, the projection is that there is going to be a lot of genetic data generated in this decade. So, the NHGRI, which is in the US, estimates that we will generate something anywhere between 2 to 4 exabytes of genomic data by 2030. And exabytes is here. So we're used to million, billion, but this is 1 into 10 power 18. And that's the volume of genomic data that will be generated by the end of this decade. And this is really vast. Like, we can't imagine how much, how big this kind of scale is. Like, for a lot of people, people say that, like, if you think of 1 million seconds, it's actually something like 10 days. But if you think of 1 billion seconds, it's some 350 years. So that's the kind of difference. You're talking from million to billion. But now think of so many more, like exabyte. So this is a lot of data that will be generated, which means that we need better algorithms to understand what this data is saying. And the idea is, can AI help in this process, right? So what is AI or anything? So I've seen this slide earlier, which sort of simplifies. It's an oversimplification, I understand. But to reason about what AI is, basically, you can think of in a traditional programming approach, what you usually have is an input data. And you will write a program that will process this data in specific ways to get some kind of output. So that's traditional programming. In machine learning or AI-driven approaches, what happens is that you have the input and you have the expected output. You give both of them to a computer, and the computer now writes a sort of a program to map this input to output. And basically, it figures out what is the program needed to convert that input to output. And then you can take that program and apply to new data and hopefully get the expected output. So that's how AI or ML can be reasoned about. Okay, with that introduction, now let's go into how AI is helping in various things that we discussed so far. So like I said, the first one is in how it is improving sequencing itself. So I told you how sequencing technologies have massively improved over time. So one of the new methods of sequencing is something called as nanopore sequencing. So in nanopore sequencing, what happens is that, so Sanger sequencing was basically working base by base, where you get a colored fluorophore emitted every time, and based on the color, you are decoding what base it is. So it's base by base. In nanopore sequencing, what happens is that this DNA, so this DNA strand goes through a biological nanopore. This nanopore is actually an ion channel protein, which is embedded in a membrane. And this DNA actually goes through that nanopore into the other side of the membrane. And there is an electrical signal that is actually present in this barrel. And as the DNA is going through, what it does is it disturbs this electrical signal. So it creates signal patterns like this. Okay. And so normally you would have a straight line, but as the DNA is passing through, it will create this something called as squiggles or disturbances in electrical patterns. And this electrical signal can be converted into the corresponding DNA sequence. So that's the basic principle of it. And previously, Nanopore was using something called as a hidden Markov model to decode this electrical signal into DNA. But now they have replaced it with a deep learning or a neural network based method, where it can take all these electrical patterns like this and use a bidirectional neural network and convert it back to the corresponding DNA sequence. And people have they've shown that the accuracy from hidden Markov model, which was 90%, they could improve it to 99%. So it became 10 times better just by using a deep learning based approach compared to a traditional statistical model such as HMM. So this means that the quality of the sequencing data you has been improved by applying AI here. Okay, so that's one thing. There is this slide that I've put for a simple purpose that whenever we sequence, we don't sequence the entire chromosome in one go because it's too long. Like you might have heard of this comparison that each cell has DNA which is 2 meters long, right? Because it's your 3 billion bases is organized in 23 pairs of chromosome, and each chromosome is 2 megabases long. So you can't sequence all of it in one go. So what we usually do is we take the DNA and we fragment it into small pieces, and then we sequence and put it back together using a computer. Okay. So the main take home from this slide is that we sequence DNA in small pieces. Okay. So once you get those small pieces, what you do is you compare it to a gold standard or a reference to see where the differences are present. And that looks, in an example, it looks something like this. So if you have a reference sequence like this, which is, let's say, from the Human Genome Project, and these are all the data points from your sample. So you map them to like that so that you see where they are matching. Okay. And like you can see, you don't match it just once, you match it multiple times. So to improve the confidence, right? So if you see for this G, for example, the data for this G is coming from 10 individual pieces. And the 10 is called, so the number of reads that map to a given location is called as depth of sequencing. So here it is 10x depth because there are 10 reads which are mapping to the G, and they all agree that it's a G. So we take this kind of a consensus approach. Using that, this is how we can visualize that you can have a SNP or a single nucleotide polymorphism, where in the reference it's a C, but in the sample it has become a T, right? All the reads agree that it's a T. And this is known as a homozygous SNP, which means that, so all of you know that there are two alleles from, there are two copies of each chromosome in you, one from the mother, one from the father. So both of the alleles show that it's a T, as opposed to this, which is a heterozygous mutation or a variation, where so in reference it's a G, and in half your data it's a G, and in half the data it's a, which means that probably one of your parental alleles, let's say the maternal allele, is the same as reference, whereas the paternal allele is a G2A substitution. And you can have other forms of variation like deletions and insertions. So deletion means that a base which was supposed to be there is not there in your data, or an insertion, which means that a base which was not there in the reference is now present in your data. So these are the kind of variations that you can study. And all this happens once you reach this step. So in a traditional computer approach, what you do is you count each of these things. Like for us, it's visually very clear, okay, there is this a deletion or there is this insertion. But what computer does is it looks at how many data points are there, how many of them are saying it's a G to T, how many of them are saying it's a G to A, all that, and it gives some score. What people have done is they've used a deep learning approach to look at images like this and understand what variation is present directly. So that's called as deep variant, which is basically variant identification. So all these changes, the C to T, G to A, this deletion, all these are known as variations or variants. Okay. So people don't use the term mutation anymore because mutation somehow gives us the impression that it's somehow harmful. But there is a lot of natural variation present, which might even be beneficial or doing anything, right? So that's why the harsh term mutation is not being used anymore. Instead, we use the term variant or variation. Okay. So what essentially it does is it performs these kind of steps, takes images, and so this is known as read pileup, as it is. So because you are piling up all individual reads, and it takes these pileup images, and it already has a trained convolutional neural network, and from there you get this likelihoods of whether a variation is present or not, and you get the variant calls or output saying that these are the places where your sample has variations, right? And this has been shown to outperform the traditional way of calculating how many reads are there, and etc. etc. So that's how deep variant works. And they even have a specific training. Like if you are working on a new species which has a different set of GC characteristics or anything, you can provide the images or pileups and the expected genotypes, and you will have your own deep variant model that you can apply to other samples. So that's deep variant. So in addition to this, you can use convolutional neural networks to understand sequence functions. So a convolutional neural network is something that we actually see in many traditional image processing algorithms. Like if you have an input like this, it takes subsamples of images and puts it through various filters or masks and classifies it as a car or not a car. In fact, every time you fill CAPTCHA, CAPTCHA you must have all seen, right? It will say pick all bikes or all traffic lights and all that. You're helping this kind of CNN to improve, because that is where it is getting its training data from. So you're helping, saying that, okay, this is how a fire hydrant looks like, or a bicycle looks like, or a pedestrian looks like, all that. So from that, it learns features like how a traditional or a typical car or a dog or a cat or a banana, whatever it is, looks like, and it can you can build a classifier that recognizes these objects. Similarly, people have applied that kind of a logic to chromosomes. So you can take subsamples of chromosomes and you will have sequence motifs or protein binding motifs, and you can learn what is conserved or common across those motifs, and then you can have a classifying output saying whether a protein will bind there or not bind there. And as most of you know, that DNA is the blueprint of life, but proteins are the functional entities in the cell. So most functions happen because the protein recognizes DNA. Even DNA, for it to be converted to RNA, an RNA polymerase has to come and sit on the DNA, bind there, right? So protein binding is very important to study. And there are deep learning approaches which help you understand whether given sequences bind a specific protein or not, right? There is a different approach called as recurrent neural networks, right? RNNs. And that's been used quite a bit in something called natural language processing. So let's say you use Google Maps, most of us use, and you're searching for directions. Let's say how to go to Warangal, right? So you say Hyderabad to Warangal directions. What it does is it takes that phrase, Hyderabad to Warangal, and it parses that into like location, direction, and so on. That's how it understands what you're asking. So this is called as natural language processing. So you are speaking in the language that you understand, and converting it, the computer is converting it into a language that it understands, right? So that's natural language processing. And these recurrent neural networks are used quite a bit in that. So people have taken the same approach and applied it to gene sequences to understand functional units of genes known as exons or introns. Right. So if you've taken some MolBio course, you know that genes are not continuous segments. There are coding regions known as exons and gaps in between called as introns. So, and coding regions, of course, will contribute to protein, whereas introns are mostly regulatory in nature, right? So you can demarcate exons and introns by applying techniques of natural language processing, such as recurrent neural networks, and understand where the gene demarcations exist. So that's about sequencing and sequence analysis. Now we'll look a bit about how AI is helping in cancer genomics, right? So most of us know what cancer is. It's uncontrolled cell growth. So usually you have checkpoints which stop cells from dividing continuously. But when those checkpoints are somehow not working, then you have uncontrolled cell growth, and that's the mass of cells that we call as cancer or a tumor. Right? Now, what happens is, which many of you might know, but these cells also die, right? And when they die, fragments of their DNA, which were present inside the cell, end up in the bloodstream, and they're circulating all over it. So this, in fact, it happens for normal cells also. It is not specific to cancer cells. So this DNA, which is just floating randomly in your bloodstream, is known as cell-free DNA. Okay, cell-free DNA, because it's not contained in a cell, but instead it is flowing across in. Okay, so traditionally, if you have to have a cancer diagnosis, you go to radiology or biopsy. Those are the methods that are, so where you have to cite the sample the site of cancer, right? Like if you have, let's say, a liver is the suspected, or lung or something, you have to take a biopsy from that area, which is very invasive because not all organs are directly accessible, right? You have to sometimes even cut open, or it's a very invasive procedure. Whereas cell-free DNA can be identified from blood itself. So there has been a lot of, because it's circulating in your bloodstream, you can draw blood from anywhere. You could argue that blood drawing is also invasive, but it is much less invasive compared to a biopsy, right? So a lot of people have been using this kind of an approach to detect cancers. So one such study, what they did is that they have actually taken cell-free DNA from random samples, both healthy as well as cancer patients. Okay, and they have identified, or they've taken the cell-free DNA from these samples and they've sequenced it. And what they do is, based on the positions of the genome where these, did you have a question or? All right, okay. So based on the positions where these cell-free DNA are mapped, they trained a machine learning model, which first of all predicts whether it's a cancer sample or a healthy sample. And even if it's a cancer sample, they could also predict with pretty good accuracy what kind of cancer it is coming from, based on where the positions of this cell-free coming from. So compared to a very invasive approach such as biopsy or radiology, even radiology, although it's not invasive, you are exposed to a lot of radiation. Like CT scans expose you to a lot of radiation, right? So whereas they could identify what cancer it is or whether it is cancer or not, just based on where the cell-free DNA is mapping. Okay. The other thing is, the other work that I want to highlight is also how a deep learning method is helping understand cancer evolution. Okay, what is cancer evolution? So let's look at a traditional scenario. So what happens is, every person goes to a hospital independently and they get sampled there. So it could be that, so let's say we're only dealing with lung cancer. So in this example, it says lung cancer, but there could be subtypes of lung cancer, right? Some are very aggressive lung cancers. Some are not so aggressive lung cancers, right? But because we are sampling every person independently, and they could also be sampled at different points, even if there are similarities between two different samples, we will not identify them because we are not looking at it in the context of everyone else, right? So in this example, this and this are very similar cancers, but one was sampled earlier, one was sampled later, because of which they're different already. But it's just that this will also progress into this if it was sampled later. Similarly, here also there are three samples which are all belonging to another cluster, but they're all sampled differently. So you think that they are different, but it's just that their sampling was different, but they would ultimately follow the same track, right? So to understand this, and why is it important to understand? If you know what is the trajectory or evolution that cancer is going to have, you can actually come up with better treatment regimens. So if you know that a cancer which is somewhere along the path is on the way to become something aggressive, then you will take a more aggressive treatment option. Whereas if you know that it is likely to evolve into something benign, you will take a more benign treatment option, right? So what they did in this study is that they've taken all such samples and have used a deep learning method to cluster them into paths that these cancers have taken. Okay. So because of this, now any new sample that you identify, it doesn't have to be analyzed independently. It can be analyzed in the context of what is already known in, let's say, lung cancers, and then you can assign a subtype to it more accurately, which means you can come up with better treatment regimens or whatever. Okay. So these two are the examples that I chose to highlight, but there is a field that is now developing called as precision oncology. Okay. So in precision oncology, the idea is that you are covering various aspects, and in each of these steps, AI is helping. So we already saw how diagnosis is improved, right? Whether a person has cancer, and if it is a cancer, what kind of cancer it is. And once you identify that a person has cancer, what is their prognosis? Is it likely to stay benign? Is it likely to metastasize? Is it going to be very aggressive, very fast, or it's going to be slow growing? And if what is the overall survival they'll have once the cancer is diagnosed, what stage it is, etc. Now, once that prognosis is identified, what is the treatment they should go through. So there's a subfield of genomics called pharmacogenomics, which I'll briefly touch upon later. So basically, it comes up with the fact that everyone responds to treatments differently. In fact, we all know that, right? So for some people, you will have better response, some people will not. So pharmacogenomics tells us what should be the dose of the treatment that a person should receive. Then what is the best possible treatment that they can have, and if this cancer is likely to respond to this treatment, or they should be ready for other kinds of treatments, and whether is the treatment the cancer is responding to it, or is it relapsing, etc., etc. And also the risk assessment itself. So even before you diagnose, so is the person likely to develop cancer, or if someone has developed cancer and it's been treated, is it likely to relapse? All those things. So in each of these steps, people have come up with AI-based tools that improve this accuracy of these kind of predictions. Okay. So the final section that I'll talk about is in clinical genomics. So cancer genomics I chose because technically cancer genomics falls as a subset of clinical genomics, but there's so much, like the burden of so high that it deserved a separate section in itself. And I think all of you can appreciate that fact. But clinical genomics basically is how genomics is helpful in studying other disorders, right? Like such as genetic disorders. So the first one that I want to highlight is something called Deep Gestalt or Deep Gestalt. I'm not sure how it's pronounced, but what this does is it takes images or face images of people, and from the face itself it tells you what likely genetic disorder they have. So again, it is a convolutional neural network based approach where it subsamples the face images, you apply a series of masks and gives you the most likely output of what syndrome it is having. So not just this, they actually made composite images of different genes responsible for these disorders. So not only does it tell you what syndrome it is, but it even gives you a good estimation of what gene mutation was underlying to cause this syndrome. Okay, so that's the kind of thing that they could do just using deep learning. Like a regular clinician probably will never be able to tell apart these subtle differences in faces where you could identify the underlying gene responsible for it. But the deep learning looks at features that a normal eye misses, and they're able to get this, right? And they've achieved something like 91% accuracy on this. So then, like I said, there is something called pharmacogenomics, where it's being used in clinics more and more now. The idea is that each of us have sequence variations, that particularly mitochondrial variations. So as you know, mitochondria is where most of these drugs are metabolized or processed, right? So there are a lot of sequence variations which makes us different in how fast we can metabolize a drug. So even if you take something simple, let's say for example paracetamol, okay? If you just, all of us take paracetamol, but for some people you will start seeing the effects pretty much immediately, let's say in 15-20 minutes. Whereas some people might only start seeing it in 2 hours, right? In some people, you will only see the expected, which is reduction of body temperature. But in some people, there might be side effects, and it's not specific to paracetamol. It can be specific to many drugs. So you will have, so if you take an average population, you will have people where you get the desired response, where you will there will be a subset where there will be side effects, and where there will be delayed or no response. So which means that so far our pharma or drugs are designed to work on the average person, but it doesn't have to be that way. If you know what kind of sequence variation is present in a person and what effect it might have on drug metabolism, we can actually come up with dosages or even medicines specific to that group. I'm not saying it has to be unique for every person, but it can be divided. It doesn't have to be one treatment for all. It can be subsets, like you have cluster A, B, C, and depending on which cluster you fall into, you will be given a specific dose or specific, like if paracetamol doesn't work, you give an alternative, something like that. Okay, so that's pharmacogenomics. And this can be also, like I said, in precision oncology, this can also be applied to treating tumors where depending on how sensitive the tumor is and your own pharmacokinetics or germline mutations which make you susceptible or respond to drugs better, you can come up with the appropriate dose and treatment that will work best for you. Okay. So there are a few other models that I have not really covered, but I thought I'll just mention them. So very recently, so like I said, you can have variations which can be deleterious, which means that they'll have some effect. They can be neutral or they can be beneficial. And in most cases, so if that such a variation is present in a protein coding gene, it's many times it's straightforward to understand the function, but not all of these variations can be or their consequences can be understood easily. And this what this ESM1B does is it created something called a protein language model. Most of you would have heard of ChatGPT now, right? So ChatGPT uses a newer form of artificial intelligence called as large language models. What it means is that it's been trained on a very large amount of English text. That's why it speaks like a real person. It understands what you're saying well, and all that. It's because it's been trained on. It's a large language model. It has been trained on, I think, 4 billion tokens of English language or something. So what these guys have done is they have done a large language model, but using protein as the alphabet, protein sequences as the alphabet, rather than English, right? So they've used some 650 million tokens of protein sequences, and based on that, now they're able to predict this sequence variation, the effect of sequence variation better. So that's this one. Then there was something called primate AI, which used sequence variation all the way from like macaques, which are the earliest primates, to humans. And they used this basic logic that any deleterious variant or which is harmful for it will evolve out, right? It will not stay in populations because it is deleterious. Whereas those which are important or beneficial will be retained or across evolution. So using this sequence conservation approach, they had come up with a AI framework where if you put in a new mutation, it tells you based on this conservation whether or not it's important or beneficial for humans. And then many of you know what CRISPR-Cas9 is. It's a gene editing based approach, which of course had won Nobel who have discovered it, and there's a lot of applications that's being out there to do gene editing. And
let's stop sharing.
From which one?
From Teams.
No, as in from which slide?
That's okay. I mean, you
yeah.
Where you are. Voice was not.
Okay. Are they visible now? Anyway. Yeah. So there is now a deep learning framework where you can design your guide RNAs which target your genome for CRISPR-Cas9 editing and predict the efficacy of it without having to test it in an experiment. Okay, so these are few that I chose to mention in the talk. So like I said, let's very briefly now discuss what are the challenges in this kind of approach. So far I told you the good part of AI in genomics, but not everything is a walk in the park, right? So the main concern with AI models is something called as interpretability. So so you might have heard that AI models are called black boxes because you don't understand what's happening. Like if you look at the sort of analogy that I gave you, where you're giving the input and output, and the computer is figuring out what the program should be. Now, what is that program? You really do not have a very good idea, and that's why it's called as a black box. In fact, I remember that when this image processing or AI for image processing was new, there was this one particular AI bot which could look at a chest X-ray and tell what gender the patient is, which even radiologists cannot. Okay, and they didn't understand how it is able to do. And later on, they realized after exploring everything, because you usually have the gender mentioned on the top of the X-ray sheet itself. So, and it was looking at that M or F mentioned there, and it was so. And these people thought it was doing it just from the lung X-ray, so they were all confused. So, because you don't understand what features it is looking at, it can really lead to misleading results, right? So that's why now they're going towards something called as explainable or conversational AI systems, where once it gives a prediction, you can ask like what was the prediction based on, and it tells you that these were the features it chose, and then a human can keep cross-checking whether it's using appropriate features or is it using something like this where some text is written on. So that's one. Interpretability is one big issue with it. Then there are regulatory and ethical issues, because a lot of data of us, and particularly healthcare records, is being used for training these data sets. So, which means that our privacy might be at risk. So that's one big issue. And whether, yes, we have gone to a hospital and given our radiology report, but have we actually given consent for that to be used in an AI system? So many times you probably aren't even aware that your data is being used for this kind of training data. So this consent issues have become a big thing. And finally, which I think is a much more question for which there is no straight answer, is the liability itself. If a doctor makes a wrong diagnosis or a mistake in saying or in giving that prediction, they are held accountable, right? Because we can go back and say that how could and whatever. But if an AI makes a mistake, who holds the liability? If you make the people who developed the AI liable, then no one will be taking the risk of developing new models, because then there will always be like no AI, even humans, they're not perfect. There will be errors, but who holds the accountability or liability for errors? So that's a big question that there is no straightforward answer to. And finally, there is bias, and this bias can come pretty much for different reasons. Like the data itself could be biased. Okay, the deep gestalt that I showed earlier, which was identifying syndromes from face images. So they had 91% accuracy in clinical data, but when this same tool was taken and used, so they tested it on European data, but if we take the same data and used it on African people, the accuracy dropped to 38%. It's because their training data was only European data, so it was performing well there. And if you give it other kind of data, then it was not performing. So that's why it's important to have diversity and have as diverse data as possible, which is representative of the global population rather than one particular subset. And then there could be like a data from coming from what is available to itself. So let's say you're trying to correlate one disease phenotype and you have 10 clinical parameters. Now, it's possible that it's a 11th parameter that actually explains your phenotype best, but you have not collected it. So what your AI is trying to do is trying to make the best out of the 10 information that it has, and it'll output something, but it may not be directly correlating with your phenotype anyway. So this is also important to see whether you're giving the correct relevant data for training or not, or you're just overfitting what you have to a phenotype that it doesn't belong. Okay, so with these thoughts, I'll stop and I'll be happy to take any questions that you have. Yeah, yeah. And also online audience, if you have any questions. I hope my slides were visible later on. It stayed stuck. On which slide? Oops. Any questions?
We're talking among each other.
Yeah, you can ask me instead. You will not be held accountable for questions. Yes.
You come on this way to talk to them already.
Should I ask you questions? Then you should be having questions. I didn't explain so many things.
Biopsy, right? So they did talk about biopsy in cancer and how newer tools are coming in. So what did you feel about that? Diagnostic. Right. It will help, but at the same time, I mean, you are all life science students, which means that tomorrow you will become life science professionals. So, what do you feel about how are you going to train yourself in using such tools?
Okay, that is a complicated question for me because I'm still a learner.
We're all learners.
Yeah, as he explained about the new invention like biopsy, it will help many people around the world. And I think not only biopsy, it should be further inventions that will talk.
Yeah. Feel free to talk. Yes.
Yeah, before that, I have mentioned, sir mentioned that Frederick, father of chemistry in 19, got Nobel Prize in 1980.
Yeah.
Biopsy, the process which converts. People get hope to diagnosis with the cancer to treat for many of us. Genome and new way to genomics, the sample and collection of tissues which color samples and they can surgeons or doctors may help to collect the samples through color where affected people. They affect the people they can conserve to may helpful to treat like to to live a simple life without any observe without any fear or
Yeah.
without harmful effects like taking medication, diagnosis, treatment, well developed treatment with the help of biopsy and AI. Some tools are helpful in AI.
Yeah. At the same time, I mean, I would also like to understand, given that all of you are students of life science and AI tools are taking over life sciences at an enormous pace, but do you also think that students should start looking at training themselves in AI tools?
Yeah. So at the very least, you should know what is the landscape of AI, like where all AI is being applied. Because yes, traditionally we have been doing things in a certain way, but you should know what newer methods are available so that later on, if you ever need to apply that for your own work or own research, or even let's say you become a healthcare professional, you should know that this option is there. Like maybe, hopefully that. Let's say you are advising someone for cancer diagnosis now, you will say, and if they are going for biopsy, maybe you will remember at that time that okay, there is also an approach of cell-free DNA rather than doing cancer. Or later on, if you are doing some work in let's say genetic disease diagnostics, you may not do a traditional PCR, and you might instead decide to do a sequencing based diagnosis. So those are the kind of things, if you're aware, you can use it in later on whatever you're doing. And if you are really interested in making your own new AI based approach for something, it's always helpful to understand how these things work. Like most of the slides that I showed, they take some training data, and so that's what is important. So you will start thinking in that way, like how to collect that training data, etc. So those are, for now, those are the basic things that you should start with, and later on you can upskill in developing your own models. Yeah.
You also have some teachers at UNP. So, do you think that the students can get these kinds of trainings in colleges or outside colleges? So there was one question from YouTube, but I also realized that they misunderstood something. I'll read their question.
Okay, which I'll clarify.
Yeah. So they ask, what are going to be the implications of AI in enhancing the genomes of neural cells in human beings? Will they surpass the ability to learn gradually in their life, or more side effects rather? And I think that they misunderstood the neural network that we discussed.
I'll cover both bases anyway. Yeah. So to clarify, neural networks are artificial, which they are just inspired by the connections that our brain cells form to like when we look at an image, it's not a single neuron that is processing. Each part of the image is processed by a bunch of neurons, and together it's interpreted by brain. So people have come up with computer vision or image-based processing in that kind of an architecture. Okay, but they're all these neurons that we talk in neural network is artificial. So once that is out of the way, the second thing is neural cells have the same genome as rest of your cells. They don't have their own genome. Okay, they have the same 3 billion bases that is present in rest of your body as well. Okay, so there is no way to specifically. In fact, it's also ethically forbidden to change genomes of certain cells in the hopes of getting some better abilities or whatever. So that's ethically out of question. And second, other thing is, you might be changing some genome thinking that it will do this, but it will also have a lot of other side effects, which the comment also points out that you might not foresee. So that's the idea.
Explain biopsy process. How do diagnose?
Okay, so biopsy, essentially what they do is, if you suspect a specific type of cancer, let's say lung cancer, you take a piece of lung. Okay, so you use a aspirational needle. So from behind, you can put it to the lung, chop off a tiny bit of piece, pull it out. It can be any. If you're suspecting lung cancer, you will do it there. Any piece of tissue, and then you stain it with something called as H&E. Full form, I don't recall, but yeah, but you do a staining, and what happens is cancer cells absorb that stain differently compared to normal cells. So based on that, you can identify whether there is cancer or not. But because of this, it's a limitation because not all tissues can be biopsied. Like if you have pancreatic cancer, it's first of all so tiny, and second, it's deep inside, which means it's very difficult to get a piece of it out. And in roughly 15 to 20% of the cases, you need to do biopsy twice to even confirm. And because it's also so small, you can, by the time you detect, it's often in stage 3 or later, where the only option for them is to remove it. And many times, even the surgical removal is not possible. Okay, which is why pancreatic cancer has very high mortality rates. Whereas if you're using an alternative like cell-free DNA, even pancreatic cancer can be identified early enough that you can have viable treatment options rather than surgery or death. Right. So that's the idea. Yeah.
Cancer, genomics, or AI.
All right. If there's nothing else, thank you all for. Yeah. Yes. Liver infection. I'm not qualified enough to do that.
You need to talk to a person who's an expert in liver infection.
Yes. Yeah. All right. Yeah. Okay. Thank you then. Yeah.
Thank you very much, Paige. And also thank you to our audience sitting here in CCMB as well as in the HRDG building and those who have joined us today on YouTube online. And I'll just recapitulate again for the audience who joined us later that this talk was a part of an ongoing talk series which is called as Futuristic Fridays, AI in Biology and Health. And what we have been doing is across the month of April, every Friday, we have an expert who is working with AI tools in any aspect of health or biology. to tell us what kind of work they are doing, what kinds of tools they are using, what kinds of questions they are solving using series. And today, Tate's talk was the third in that series. Next Friday will be a final talk in the series where we will be meeting Dr. Anshu Bharadwaj from CSR Intech in Chandigarh. And previously, we have met Dr. Rajan Rao from University of Michigan, who uses AI tools for imaging, like Tate was talking about X-ray diagnostics today, right? Like, so there are many kinds of imaging tools that are used in healthcare. And how are we using AI tools for understanding images better? And then we also met Dr. Lipi Thukral, who's from CSIR Institute in Delhi, called as IGIU. And she talked about how AI tools are being used to understand structures of proteins, like millions of proteins that make our cells. How do we understand those structures using AI tools? So I will urge all the students who have joined us today in person or online to take a look at our Futuristic Friday series under Jigyasa. And Jigyasa is our flagship program under by CSIR to connect scientists and students. And we connect school and college students with scientists in various fashions. For example, some of you who have come to the Research Institute today is also being covered under Jigyasa, or having these kinds of talk series, as well as we have a website called as Jigyasa, which has loads of content that can be used in classroom settings. It contains content in form of simulations that perhaps colleges will find useful, schools will find useful. There are many experiments that we do in real labs, but are probably not feasible in your school and college setups. So we have made those simulations for you to use. There are other content in form of videos or comics, or posters that educators should definitely take a note of and use in their classrooms. With that, I'll close the session for today. Thank you all for joining, and hope to see you in person or online next week again. Thank you.